Okay, so we move to the third presentation of this last heat. We have Dr. Kasra Khalaj representing AAP with human amniotic fluid stem cell extracellular vesicles rescue features of lung development in human hypoplastic fetal lungs. So let's see it.
Thank you for the opportunity to present our work. It is well known that the lungs of babies with pulmonary hyperplasia, secondary to congenital diaphragmatic hernia, have the impaired triad of factors with impaired fetal lung growth, maturation, and vascularization. Although some therapies have been administered prenatally to treat pulmonary hyperplasia, none of these have been able to fully rescue lung development. In search for a regenerative therapy that could rescue normal lung development, we started studying stem cell-derived extracellular vesicles, which are particles responsible for paracrine signaling, and in particular, those from amniotic fluid stem cells, or AFSCEVs. We have reported the main results of our studies using AFSCEVs on several rodent models of pulmonary hyperplasia. Now, we have been investigating for the first time in the human and understanding the clinical relevance in human pulmonary hyperplasia. I'm excited to show you all a novel human fetal lung explant model which we've now established at SickKids. Fetal lung samples were harvested from terminations of healthy fetuses at 15 to 19 weeks of gestation, and these specimens were cultured as explants and treated with medium alone for the control group, or an inhibitor of RAC1, which is a Rot GTPase that modulates lung branching morphogenesis to induce pulmonary hyperplasia. We have confirmed that RAC1 expression is downregulated using CDH autopsy specimens compared to age-matched control. We have now switched to human-derived amniotic fluid stem cells and characterized these EVs for size by nanoparticle tracking analysis, morphology using transmission electron microscopy, and Western blot for canonical surface markers following the International Society for Extracellular Vesicles guidelines. We've assessed the triad of dysregulated factors in pulmonary hyperplasia through the following outcome measures phenotypically, and we also investigated the changes induced by AFSCEVs at a cellular level by performing single nucleus RNA sequencing on all three of our groups using the 10x platform. I'll first walk you through our phenotype data. Here you can see encouragingly that fetal lungs treated with the RAC1 inhibitor display histological features resembling those of pulmonary hyperplasia. And when we administered the human AFSCEVs, we saw a restoration of airspace density with decreased parenchyma as well as branching morphogenesis marker FGF10. We also see a restoration of key markers responsible for fetal lung maturation, surfactant protein C and podoplanin, when we administer human AFSCEVs to these hypoplastic fetal lungs. When we looked at the vessels, we found that human AFSCEV-treated explants had reduced medial wall thickness and restored vascular density. Encouraged by our phenotypic findings, we then wanted to understand the effects of this treatment that were being exerted on hypoplastic fetal lungs at the single cell level. With single nucleus RNA sequencing, we built an atlas of pulmonary hyperplasia, and here you can see a UMAP identifying all major cell types comprising of the fetal lung. Specifically, when we look at the epithelial cell populations in red and subcluster them, we found a unique group that we call the stressed AT1-AT2 that had high representation in hypoplastic lungs that was downregulated after AFSCEV administration. This cluster had upregulated signaling pathways involved in cellular stress, death, and inflammation. We then looked at a cluster we called developing AT1-AT2 cells, which have the highest proportion in hypoplastic lungs treated with AFSCEVs. This cluster had high TGF-beta and VEGF signaling. When we look at the fetal lung mesenchyme, we see that human AFSCEV administration reduces the number of myofibroblasts in hypoplastic fetal lungs. And interestingly, in the endothelium, we see that human AFSCEV administration can restore nuclei numbers back to control in this cluster at the top, the vascular endothelial cluster, which is consistent with our phenotypic findings. And this cluster had increased VEGF signaling, which is a primary pathway involved in angiogenesis. So in conclusion, we are very excited as it is the first study to describe that human AFSCEV administration promotes the triad of factors: fetal lung growth, maturation, and vascularization in fetal hypoplastic lungs. And so moving forward, these results have direct clinical relevance as an AFSEV treatment represents a promising cell-free approach to improving lung growth in babies with CDH. Thank you to our funding sources, and thank you for listening.
Awesome. Welcome, Dr. Khalaj. Thank you for this presentation. I have one question because this seemed really promising, but my question is, what do we need to move this to the real clinic patient? Like, how will we administer this and everything?
Thank you very much for that question. It's one we get all the time. And I'm really excited to share with you. We've actually started doing some work with the same source of these extracellular vesicles in a lamb model of CDH. And this is in collaboration also in Belgium, but we've got our own colony that we are working at the University of Guelph here in Ontario. And so we've been testing it so far preclinically, and so this is obviously going to answer that question. And so we're also applying for further grant funding to support further studies in this lamb model in order for us to really get this to the babies with CDH.
Awesome. We have a question from the audience that Soo-Jong Kim. It's in practice, will the stem cells be injected into the amniotic fluid?
So that's a really great question. Thank you very much for asking that. We've tested various models, routes of administration. We tried intratracheal in our animal models, I should qualify, through maternal IV, etc. So far, we've had a lot of promising results with the intraamniotic injection. The ones that we're doing with the lamb model are in combination with fetal, so they're administered in combination with fetal. And so this would be a combinatorial-based approach so far. But thank you very much for that question.
Awesome. Great.
Phenomenal work.
Thank you.